Gracias por asistir el día de hoy a esta conferencia de jueves de la Universidad. Como todos ustedes saben, tenemos cada jueves en el Instituto de Ciencias Naturales. El día de hoy tenemos una invitada muy especial que nos va a hablar sobre lagartos, Antillean Anolis, que muchos de ustedes me parece ya conocen. Katharina WOLLENBERG hizo su diplomado, bueno, ella es de Alemania, está visitando el país, lleva acá algunos meses, por razones familiares, pero también porque tiene un interés muy especial en trabajar con los anolis de la Sierra Nevada de Santa Marta. Katharina es doctorada de la Universidad Técnica de Brunswick. Su tema de doctorado no fue en anolis, pero igual es un tema extremadamente interesante. Fue sobre sistemática, patrones de diversidad y modos de diversificación y radiación de anfibios endémicos de Madagascar. Actualmente es investigadora de la Universidad.Department en Alemania. Tiene pues, una lista impresionante de áreas de interés. Entre ellas voy a mencionar solo algunas: desarrollo de adaptaciones ante el cambio climático, desarrollo larvario de anfibios y reptiles, genética del paisaje, geografía de la conservación, genómica de poblaciones y otras. De hecho, su interés particular en la Sierra Nevada de Santa Marta involucra este aspecto de evaluación de posibles adaptaciones ante escenarios de cambio climático. Entonces, Katharina nos va a hablar el día de hoy sobre, bueno, el título de su charla en español es Explicando la diversidad al interior de la radiación adaptativa, tomando como ejemplo los lagartos anolis de las Antillas Mayores. La conferencia va a ser en inglés, acuérdense que al final hay espacio para preguntas. ¿Ok? Katharina, you're welcome.
I think we have to start. All right, I'm going to start talking anyway. So, yeah, it's nice that you're all here. Nice to see you. I'm going to talk today about adaptive radiations, not only about the Greater Antillean Anolis lizards, but also other examples. Among those, the frogs that I've been working with in my doctoral thesis, because it all fits very nicely together. The work on Anolis that I'm presenting, I did a postdoc at the University of Harvard in the lab of Jonathan Losos, who is quite famous for his work on Anolis lizard radiation. So that's what I'm going to present you today. First of all, I'd like to explain you what is an adaptive radiation. You probably know that already, but an adaptive radiation is characterized when a generalist ancestral species radiates into many descendant species by ecological specialization. So we have a generalist ancestor, like here in the example of birds, and then we have a very fast burst of speciation and many different specializations. So the rapidity of the whole thing is a characteristic. The diversity that results is another characteristic. And if you do a DNA phylogeny, most often what you're going to get is some sort of starburst radiation. This is an example of the African HIV virus. And let's see if this works now. It does. And that's another example that's been published, I think, today in Science. That's a phylogenetic tree of all the birds of the world. And actually, let's see, next slide. And actually, the interesting thing is that usually in adaptive radiations, after this initial colonization and the diversification into open niches, usually at some point the speciation process slows down because all the niches are occupied, and that means by decreasing ecological opportunity, just the rate of speciation decreasing. The problem is in birds that this is not the case. Today in the paper I saw that they found that the speciation rate is ever increasing. So we don't know why. This diversification is by mostly divergent selection and niche partitioning. So you can see there's like some sort of functional types, and they're going to evolve according to different functions of feeding, for example, like in the bird beaks, or other morphological adaptations. And the question is, if you have a population that's growing and it has like disruptive selection going on, why is that population diverging into two? And for that one, we have to look at the geographical mode of the whole thing. So there's the two classic options, that is allopatric speciation or sympatric speciation. Allopatric speciation being thought by the founders of the new evolutionary synthesis to be the prevalent mode. So basically, if you have a population that wants to be two new species, you need a geographic barrier, some sort of barrier. It needs to be either a solid barrier like islands, for example, or a soft barrier like, for example, mountain range. And the other option is ecological speciation, where genetic polymorphism is under disruptive selection, or alternatively is autotrophic mating, that would be sexual selection. And in the end, you have two populations arising at the same place. That's not very often, how you say, validated, or the concept is not very valid at the moment still, but there's increasingly more examples that this is also taking place quite frequently. So I'm going to show you some examples of adaptive radiations. In this talk, I make a distinction in two types or stages. Types, I say, because sometimes it's only one of those, and stages it could be if both types are present. The first one would be diversification among islands. It could be also any other type of habitat, but I study islands, and I'm going to show examples of islands, so in this case, islands. The first one is an ancestral species comes and radiates into different islands, so you have allopatric speciation. It's very easy to explain. On every island we have a different new species sitting in the end. The other one would be diversification within islands, which is a little bit more difficult to explain, but I'm also going to show several examples, and also my work is mainly centered on those. So for the first type, that is the diversification among islands, you have speciation that follows ecological specialization in allopatric, and the first driver of this is, of course, the subdivision into different populations by those barriers. And also, then you have differential selection or genetic variation of those populations, and also sometimes you have the effect of random processes such as founder effects, which basically means that the descendant populations are non-random peaks of the founder population, and also genetic drift, for example. So the most known example for adaptive radiation are, of course, the Darwin's finches. If you see the Galapagos archipelago up here, you have on each of those islands some sort of ecological specialization of an ancestral type of bird into different morphologies and different habitat uses, like you have different types of beaks for different feeding types, and they have radiated into in total 14 species, which is not very much, as we're going to see right now. The next example is from Hawaii. That's the radiation of Drosophila, the picture when Drosophila flies, where you can see very nicely the pattern of allopatric speciation, because basically the colonization routes that you can infer are in the sequence of the birth of those islands. So the islands came up one after another, and the Drosophila colonized those islands while they were being built. And those are about 800 species, and they occupy a very different range of habitats as well, and also have accordingly very different morphologies. So then we have on Hawaii another example, the honey creeper birds. That's also a passerine family, also finches, and they evolved about 34 species, 13 of them are already extinct, with very different beak morphologies according to their feeding habits as well. So we have ecomorphological types, and pretty much there's a nice study that says from Losses and Wickers in 2009, they analyzed these convergences between Darwin's finches, other finches, and those Hawaiian finches, and they found that even though there are very convergent adaptations, like for example beach shapes, they occupy different spaces in morphological space. So this would be like a morphological principal component. I don't know if you heard about that before. It's like a summary variable, and you see that the Darwin's finches, the triangles are down here, and the Hawaiian ones are up here. So even though they look the same, they're not the same. Another example from lakes, also very, actually that's one of the two famous examples for adaptive radiation that people usually know, are the cichlid fishes from the Rift Valley lakes in Eastern Africa. Here's that's the rift, and basically by the drifting part of those tectonic plates, you have lakes arising. But the interesting thing is that if you look at the phylogeny of fishes from one of those lakes, those are different markers. For example, you see that different colors are different sources. So you have that radiation that basically includes different source populations, which means that it's arose by allopatric migration of different sorts of fishes into their lake. And the one I'm most interested in, the radiation of the Anolis lizards. They are distributed in circum-Caribbean and in North America up to Georgia, also in South America, which is not shown here. And also here you can see, if you look at the phylogeny that's been published last year with several nuclear genes, that you see the different colors representing the different origins. And for example, this split here between Jamaica and South America gave origin to those lineages. So also we have an allopatric speciation pattern. So let's get to the next example, the one for diversification within islands. And that's, as I said already, it's more difficult to explain. So you have different factors that can account for speciation that happens within those islands. One would be that basically there are islands within islands. So you have different sorts of habitat that count as areas for allopatric speciation. And also you have alternative explanations such as ecological speciation, or for example also life history traits. I'm going to explain later about that. So an example for this might be the cone snails. Not very much is known about them. So you have these marine snails that predatory, have a large range of morphologies, a large range of color patterns, and a large range of prey types. But they're all distributed in the tropical Pacific. So there's apparently no barrier to them, but still they managed to evolve into 500 species. And this is, for example, a haplotype network depicting this. So you also see again this typical star shape characteristic of adaptive variations. But this is, for example, still very under-researched. Another example is, again, the cichlids. So despite the fact that you have some speciation that is explicable by migration between those lakes, the most of the species diversity arose within those lakes, right? So Ernst Mayr, basically the founder of allopatric speciation, said there's a problem because obviously in those lakes there are no barriers. So how did all that happen, right? All of this diversity here. And there's been an immense body of research dedicated to this variation, and the latest idea is that basically you have characteristics of sexual selection and characteristics of natural selection that both contribute to the evolution of new species within those lakes. Because you do have barriers, you have different water depths, you have different amount of solar radiation, which can count as barriers for speciation. And on the other hand, you also have intense sexual selection by sexual dichromatism and different females preferring different males. So I just mentioned the Anolis shortly in the context of allopatric speciation, but I'm more interested in finding out what happens within those adaptive variations, within places. Why do lineages diversify? Because there's such a high species diversity, and allopatric speciation cannot account for all of this diversity. So I was trying to find out how that happens with the furs of Madagascar and with the Greater Antillean Anolis lizard. I'm going to explain a little bit about Madagascar as well, because I think it fits nicely into the story. Madagascar is a very interesting model region to study evolution because it's divided into a very simple pattern of habitats and climate. So you have humid forest, you have central highlands that are mostly dry, then you have a very dry and hot deciduous forest on the west coast, and spiny, very hot in the south. So basically, many animal groups of Madagascar show diversification patterns that kind of are common with each other, and you can compare them to find out like how evolution shaped those animals occurring there. An example is the birds of the family Noviembre. There are 22 species, and again you have this crazy diversification in beak shapes and in feeding morphology. And you don't see that well, but here's a phylogeny, and it has like little symbols, and those little symbols correspond to the little different symbols for habitat types we have here. So the beak shapes and their respective species are sorted into different categories of habitat. So basically, in this case, it means that those habitat types serve as barriers for population divergence. Another nice example is the one of Madagascar lemurs. They're very famous primates from there. And what you have here is you can see that the species are explicable by these areas here. And these areas, they're called retreat dispersion watersheds. So what happens is during climatic oscillations in the Miocene and Pliocene, we have river catchments that went like up and down in the mountains. And by those movements, isolating groups and populations of lemurs that were in between. And the thing is that lemurs are very averse to water. They don't like to swim, they don't like to cross water, they don't like to go near any water, they really don't like it. So in that case, that serves as a barrier as well. And we have all of these different species today in those endemic watersheds. So regarding the frogs, there's 3 families. One of them is very diverse. That's the Antillean with over 260 species. And you have, just as all the other animals groups, very different morphotypes, like for example these tree frogs, or we have stream frogs. Where are those? Stream frogs. And many, many more. So the question is, how's that explicable? This is all of it nicely sorted into phylogeny. So you see the Antillean are basically from here to here, of the species with the different genera, and these are 2 other ones. And so you see already, there's so many species, there can't be that many climatic regions. So what was happening there? So I'm trying to explain the whole thing with biogeography, with the different climate regions. We try to reconstruct the ancestral distribution areas of genus of those frogs, the genus Diversification. They are terrestrial leaf litter frogs, diurnal, independent from water bodies, so that's why they're interesting to study their dispersal. So we made like this analysis, coding different areas of biogeographic animism on the phylogeny. So red would be one, and yellow would be one, and blue would be one. And we saw how many transitions are between those. And we saw that basically those major clades of Diversification, they very well correspond to different areas of animism. For example, there's a clade that sits in this very hot western dry habitat, and there's a clade that sits in the northern, more humid areas, and there's a clade that sits in the southern ones. And there are not many transitions between those regions required to explain those clades. But however, the most of the species are within those clades and within those regions, and we still don't know how they got there. So we try to think about something else, like a little bit outside of the box. So how do we explain all of this? And we were thinking about maybe body size is a good indicator. It's a life history trait, and if you think of it in terms of diversification, and if you consider the fact that most frogs in Madagascar are very small, have very small distribution areas, we were thinking that maybe that's a mechanism that explains that. So we proposed a phenotype, and we call that microendemic phenotype. And under that hypothesis, we would have frogs that have small body sizes, exhibiting limited dispersal capability, so they cannot move that much, and low physiological tolerances, because you know that frogs are very strictly limited for their breeding biology. So if a frog is small and has a microclimate that is adapted to, it's probably not going to be able to migrate hundreds of kilometers to another one, because it's going to encounter many adverse habitats in the meantime. And that would lead to smaller ranges because of that, which would facilitate reproductive isolation among populations, and probably might favor speciation. So we try to test that by constructing a multi-gene phylogeny of all the Madagascar species. You see it's dated here as well. The origin of the Antillean is about 44 million years ago, and most of the species diversity that we have today is very young. We identified sister species. That's it's not very important. It's just a way of how to calculate phylogenetic independent contrast without considering the whole tree. And you see a couple of things. For example, if you plot the age of the frog sister species versus their distance to each other, you see that all of the young frog species are very close to each other, and all of the older species pairs are most likely more far away from each other. And also, you see that the distance between those frogs' ranges is also proportional to the distance difference in their range size. So new species that just arose are very close together and have very small range sizes. So that discards a priori the hypothesis of peripatric isolate speciation, which would be that you have one species that is very widely distributed and it's making basically new species along the edges, right? So this one we don't have here, otherwise we would have expected a different pattern in those frogs. So also what we found is that small frogs have a very high mitochondrial substitution rate, which means they evolve faster. So actually it's a very good proof of our hypothesis that we said that for smaller frogs, obstacles are higher. That's why they probably are so diverse, because even small areas are basically have many geographic areas. So in short, life history traits can explain some of the diversification within those regions of fungal animism that we defined before. So let's move on to Anolis. And the Anolis, as I said, they're sitting everywhere from Georgia in North America to South America, the Amazon basin. And I was focusing on the Greater Antillean. So that's this island group here on the south of Florida that encompasses Cuba, Hispaniola, Puerto Rico, and Jamaica. And most of my studies have been in the Dominican Republic because Haiti, which is on the left part of Hispaniola, is not very accessible right now. So I've been doing most of my studies on this side. Well, diversification within islands. There's a very neat story for the Anolis, which is probably the stories that most people know. So you have, basically, if you go on each of those 4 islands, on every tree you can see this type of Anolis sitting. So that's ecomorphs, they're called. Basically, it's ecology, morphology, behavior specialists. That means that, like for example, big ones always sit on the top of the trees and always eat something special or behave in a special way. Or for example, you have those very small ones that are sitting on small twigs and have short limbs and they're basically moving very slowly like chameleons. So this is basically, well, happened independently of each of those 4 islands. And you see very nicely that if you look at the phylogeny of those Anolis, and every symbol type would be one of those ecomorphs, they don't cluster together. So the crown giant from Cuba is not related to the crown giant from Hispaniola or to the crown giant from Puerto Rico, but instead in Puerto Rico this one is related to that one. Okay, so this is convergent evolution, and they're all clustering very nicely in the morphospace if you look at that. But on the other hand, if you look at the number of species that we have, there are 140. It doesn't explain that, because if you have 6 ecotypes on each island, that would be 24, right? So we're again missing a part of the picture. So the question would be, how does diversification within Anolis work? And we have, as I just said, the majority of species are within those ecomorphs. And the ecomorphs, that's another figure from Williams in 1983. You see those ecomorphs again. They're diversified mainly according to the axis of the perch. Like if you have the tree, one is going to sit up, one is going to sit here, here, and here. And the question is, like, how do we explain the rest? And Williams already had an idea. He said, oh, we have the climate axis. So basically, if you have several species of that one, that would segregate into different climates, again, allopatric speciation, but it's not very well researched. So this is what I set up to do. My study group was the trunk ground Anolis of Hispaniola of the Dominican Republic, and the idea that we had was that you can basically connect the pattern to the process. You can see how diversification happens if you compare different levels. So we compare the level of the ecomorph radiation here along the perch axis, and we look at what happens between species, and then we compare that with what happens between populations of species right now. What does that correspond to? Which pattern does it correspond to? And the group that I chose was those trunk ground anoles here. Here's a little species phylogeny, so you have the Cybotis species complex, that's what I call it. And you have the species Whitmani, Longitudinalis, Traumae, and Arcanoi. And another study found already that those segregate into different microhabitats. These ones are very highly diverse, 5 to 18% divergence in the ND2 gene, which is quite high for Anolis. So I set out to compare those populations to see if they correspond to this one or to that one. And the very underlying question of this is the question of determinism or contingency. So that's something you always have when people discuss about how evolution happens. Some people say, okay, you can predict that because it repeats itself, right? So in this case, that would mean that the diversification of my populations occurs in a manner that has already occurred before, because evolution is under the same type of selection, will always yield the same outcome. On the other hand, there's the people that argue for contingency that says every evolutionary event is unique because there are so many parameters that you have to consider that it's very unlikely that it's going to yield the same outcome. So in this case, we would expect the diversification of Anolis cybotis, species complex, to occur along axes that we haven't seen before and being unpredicted. So in this big framework, we test 2 hypotheses. that are recurrence and spatial convergence. I'm going to explain that more in detail. So the first hypothesis of recurrence basically means that if you consider the diversification of those ecomorphs in the same structural microhabitat niche, basically would expect the populations of Anolis sagotas also exhibiting that pattern. If you consider diversification within ecomorphs, aka the species of sagotoid anoles, like the different species you have ecomorph here on that island, that are segregating in different climate zones, you would also expect them to segregate into different climate zones. So that's the question. What is going to explain morphological divergence and genetic structure? Morphological divergence could be either explained by the environment, which would be this axis, or by the structural habitat, which would be that axis. And of course, you have to consider many other interactive effects as well, because of course, genetic morphology is not independent from genetic relatedness, and also the environment and genetic relatedness might work together, because different populations will be segregating different habitats. Right? That's what I just said. So we have to consider the effects that we want to, and also the ones that we don't want to. The second hypothesis was spatial convergence. That's very easy. Basically, you expect if there's deterministic evolution going on, you would expect similar ecotones or similar habitats to produce the same phenotype. And that's what we tested with the montane ecotone. So you see in red are mountains, big ones here, and then you have 3 smaller ones here that are more or less separated from those. So we already considered landscape effects. So, of course, there was some data that needed to be collected, and data was collected between -20 meters elevation in the Plan Cul-de-Sac here. And that's a low depression on the other side is Port-au-Prince, where there was the earthquake, so you have 2 different plates here. And we collected data up to 3,000 meter elevation here on Pico Duarte, that's the highest point in the Caribbean. And well, we call it GPS data, habitat use data for 551 males. We collected 251 species for osteomorphology, and also we did some modeling and added a couple more occurrence records from the museum. So this is just to show you like how it looks there and where we find those lizards and what crazy diverse habitats. So first, you have this coastal mangrove forest that's very thick, very dense, very low light conditions. You find lizards there. You find them equally in the xeric forest, where you have basically limestone, and the limestone is soaking up all the water that rains. So the vegetation is spiny acacias. You have them there. And also you have them in the montane regions of Hispaniola, where you have mostly upland pine forests and bromeliads. It's very humid, very cold, and you find them there as well. That's a female. Then, by collecting, we already saw, okay, there's some crazy amount of phenotypic variation going on here. If you alone look at the dewlap colors, which I use frequently for Anolis to study divergence, so you have like these red ones here, some red ones, very yellow ones, very white ones, and also you see already differences in head shape, like you have flat heads, thick heads, and very long heads, for example. The data that we collected was basically this data tables that I worked with them. So that was for DNA, we did ND2 barcoding in order to assign the data that we got to the studies that have been done before. For morphology, we recorded traits that are known to be adaptive in anoles. With the calipers, we took head shape measurements, and also we did X-rays and measured 53 landmarks on the bones. And also we scanned the toe pads, because toe pads are also known to be very adaptive in Anolis. For habitat use, we collected climatic data, both from our own recordings and from the bioclim layers. And also we recorded the structural microhabitat use, aka where sits the Anolis, what does it do, how does the habitat around look like. So for analyzing the data, we chose a landscape approach, which basically meant that all of those data tables that we had to be transformed into maps. And how we did that? By using the criteria algorithm in ArcGIS. So basically you have the species values and you interpolate them according to the algorithm over the landscape. Right? So we did that for morphology, for habitat use, and for genetics. There's also a possibility to do that. We added the information from the bioclimatic layers. And also, I said already, we did some habitat modeling. So we did that using Maxent, using all the occurrence points from the museum. And that was just because if you do landscape analysis, and you extract data from there, you might be extracting data from places where the species actually doesn't occur, but because you interpolated the data, you don't see that anymore. So by calculating that habitat suitability scores here, we could basically weight our analysis with this variable to basically downweight points that come from areas where there's no anolis because they're lake-ready or very dry and hot areas. Okay? And then what we did is, I cannot see anything here, so there are like 1,000 little points in there, so we extracted values from all of those maps and then did statistics with those. For the first hypothesis, if you remember this little pie chart here, we wanted to check what influences are there on morphology from our different variables and from interactive effects of those and effects on each other. And we found basically that something that is first very nice, like you see significance, yay, you know, everything's significant. But the problem is if you have everything coming out as significant, like for example, environment, the axis of diversification within ecomorphs explains perfectly the morphology of anolis. What is diversification? On the other hand, you see structural habitat variation, which would correspond to the other axis that we wanted to investigate. The diversification among ecomorphs also explains morphology very well. So in this case, what do you do? Like, which one's better? And also we found that morphology is also very highly determined by genetics, which is to be expected. And also genetic structure and environment covariate, because probably there's some sort of allopatric sorting going on. So if all predictors are significant, you have to look at the effect sizes. And that's what we did by using variance partitioning in R. And I'm going to explain that right now. So basically you look how much of the total variance or morphology you have is explained by each of the predictors you use and by the interactive effects. So what comes out is that by this model, we could explain 69% of the total variance of morphology here in the middle, where 24% is explained by genetics. So most of the morphological variance in those animals is explained by their genetic relationships. On the other hand, you also have, despite the covariation between genetic distance and environment by different plates occupying different habitats, you have a significant association between morphology and environment. It's 7% of the morphological variance, which might not sound very high, but if you consider that you have 24% already here, it might be high, you know, considering it's an independent effect from genetics. I'm going to show that in the next slide again. And 3%, small 3%, is explained by the structural habitat use. So we still have both axes being significant that we wanted to test, but now we have like a little better idea of how much exactly it is explained by those. So I just show you the 3% of structural habitat use. It's only 3%, but if you put it in a correlation, in this case, economical correlation analysis of all phenotypic variables and all variables of structural habitat use, you see that the correlation is very strong and is very significant. It doesn't matter in which way I prune my data, it still comes out like that. It's almost perfect. So what does that actually mean? That means that the phenotype corresponds to a neat shift, right? Structural habitat use, and in this case, it's a neat shift from the trunk to the ground. Right? So different populations of anolis cyanotes have a different height of perching. So we have some sort of divergent selection. You can combine that with the environment, because as you saw, environment was in there everywhere. And you see that basically this trunk ground neat shift, both of those now in here, are also explained by an underlying change in the environment. So you see still correlation very nice. All right, environment. So let's get back to hypothesis number 2, the spatial convergence. So we wanted to find out if there's evidence for spatially convergent trajectories of evolution in morphology over the same type of ecotones, and we used mountains for that, right? So the interesting thing about those mountains is that they provide an independent test, because this hypothesis has been tested in the Lesser Antilles before, in a population of anolis that inhabits a mountain. However, there was only one, so you don't know, if you can actually replicate that result. And the good thing about our system is that there's dry forest in between, and there's different genetic populations sitting on each of those mountains that are very far away from each other, and even you have like 3 little ones more that you can use for testing. That's basically what we did. So this is just for visualizing, again, the same plot that we had before. So we have the environment on one axis and the ecomorphology on the other axis. And what we did is just paint the different points from the different mountain ranges in different colors. So if you look at this, you see that basically hollow ones are the large ranges and the filled ones are the small ranges. you see that all of those mountain ranges cluster in the same corner of the ecomorph environment area. So basically all animals that are inhabiting the same type of environment have the same type of ecomorphology, as opposed to the lowland right here. So if you put that on the map and do statistics with it, you see that in each of those ecotones, if you put the if you test the morphology between lowland and each ecotone, you see that it's also always very different from the lowlands in morphology, and also it always varies in the same direction, right? So you see on every mountain the value is always lower than the ones in the lowlands, 6 times. And how does that exactly look in terms of lizard? That's pretty much that lizards in highland expose like very stocky body type, and they're perching flat on the ground, and lizards in lowlands, they're very slender, and also perch vertically mostly on plants, and those are sitting mostly on rocks and logs. So, as a conclusion from this one, you can see that the first hypothesis that we had, if the diversification of Anolis cybotis mirrors the one that we know before, it does that, and it does it in 2 instances. We wanted only one of those, but came out both are important. So basically we have divergence on this perch axis, like in the Anolis ecomorph radiation, and also we have diversification on the bioclimatic niche axis. And for the second hypothesis, we found strong landscape effects that basically if you have the same type of environment or adaptive landscape in that case, you always have the same phenotypic response. So that's a triple evidence for deterministic evolution going on here. And in terms of the radiation problem, if we have allopatry or something else, there is no sole explanation for this, because we have some evidence for this, and we have to have some evidence for this environmentally associated diversification along clients, not among areas. Right, mental switch. So we're back to ecomorphs. So right now we've been looking at populations of this guy, but there was another problem coming up, which is that if you look at the diversification within islands into those ecomorphs, like it usually is supposed to happen is that you have competition, right? So the ancestral Anolis gets a large population, and then they have to evolve in different niches, otherwise they're going to die out. However, if you go on one island, and you go on different places on those islands, you almost never find the whole picture. Like the idea is, okay, people fly to the Caribbean, they go here, they see a tree, and they see all of the Anolis. That's not the case. But sometimes there are some missing, and sometimes you have them complete, and sometimes you have none of them. So the question was, what promotes that differential community assembly? Maybe also in order to explain what explains the ecomorph evolution. So what we did for this one, we just modeled the distribution of ecomorphs. So that's like a species distribution model, but it's not with species, but it's with ecomorphs. And what we did is we overlaid those. So basically on the red areas, you would find all of the ecomorphs on one tree, and on the green areas, you probably wouldn't find any, or just one or maybe 2. So that's the completeness of ecomorphs communities, or abbreviated ECC because it's easier, right? So you see like that pattern is pretty patchy on all of those islands. So basically, it's not that if you go there, you find them, you have to look for the places where you find them. And the first thing we wanted to know is basically if the different islands, if the places where you find all of those ecomorphs are determined by the same bioclimatic niche. So you can say, okay, this bioclimatic niche, if we have these parameters on all of the islands, we're going to have ecomorph communities that are complete. So what we did is we basically modeled each of the islands with its own data, and then we repeated the model with data from the other islands. And if you would expect that it's the same climatic niche, you would expect to see the same pattern in all of those, but it's not the case. So you have, for example, in Jamaica, modeled for Jamaica, you have an equal occurrence probability of all ecomorphs over the landscape, and if you use the data from Hispaniola, it's nowhere. So you can also compare that with the niche similarity index that basically just said that those islands are all different from each other because they have very low values, or values under 0.5. The most similar ecomorphs across all of those island models are the crown giant anoles in all comparisons, and the least similar ones would be the twig anoles. So we have communities that are patchily distributed, but they're evolving in different bioclimatic niches. That's not good because now we don't know why they are where they are. So we have to find out why. So there was another idea. We correlated that with elevation. And you see that in Jamaica is the only one that falls out. So Puerto Rico would be the blue crosses here. You see like a nice correlation with elevation. Hispaniola would be the black dots. You also see a very nice correlation. Cuba has a lower R value, but it's very significant because it's a significant increase, although the values are not very high. But Jamaica completely falls flat. So because you see, if you see elevation here and ecomorph community complexity, you see basically like a hump-shaped pattern. So there's like an increase at mid-elevations, and then it falls off again. And that's a map of elevation that you can basically visually compare that as well. So apparently, whatever the climate might be, ecomorph communities are complete on mountains. Why is Jamaica different? Because we were thinking, maybe there's something else that makes Jamaica different. So we did information criterion-based model selection among different models that we had of predictor variables. So the first one we had like all of the variables, basically elevation and all bioclimatic variables, and that was the best one for Hispaniola, Cuba, and Puerto Rico. So it's not the actual environment, it's a specific environment that you find in mountains. However, on Jamaica, this is a very bad model, and instead the preferred one would be just a climate without elevation. And you see that here as an example for one of those bioclimatic variables, that's temperature range, annual and diurnal as well, it's the same. They're very different among those three islands and Jamaica. So Jamaica has basically no temperature range. And then we asked ourselves, like, why do you have that correlation with elevation? Like, what's so interesting about climate that's associated with mountains that you would find all of the ecomorphs there, but not somewhere else? So what we did is we correlated that with body size, because we have very small ecomorphs and we have very big ones, and there's an idea behind that they might potentially have different physiological properties, right, or requirements. So there's a very small one, that's a twig ecomorph on my finger here, or on a pen. And that's the correlation. So if you have the sorting of the ecomorph size classes from small to large, the small ones here and the large ones there, you see like a significant negative correlation with elevation. It's basically how stringent they're related to elevation. So that's the R of elevation and ecomorph occurrence probability. That means if you have a high R, that means that ecomorph occurrence probability is higher at high elevations, and that's occurring. That's valid for the twig anoles, but not so much for the crown giant, and that's why you get this sort of negative thingy here. And then I put two different p-values up here. So you see the Jamaica twig anoles are kind of destroying the picture, these ones. If you take them out, it becomes significant. But as I already said, Jamaica is different, and mostly the Jamaican twig anoles are falling out. So we need to find out why they're falling out. So it's because there's only one species there, it's Anolis valenciani. Maybe it isn't the twig ecomorph. So that's the ones that you usually would find like in Hispaniola, very, very small and tiny. And that's the one you find on Jamaica. It looks like a twig ecomorph, but it's huge. It's basically like 3 times the size of those. And it was even classified by Williams in 1983 as a giant twig anole. So also they perch like really high up in the trees. And it might be a different ecomorph, or on the other hand, if you put an evolutionary hypothesis to this, it could be that Jamaica produced this different type of ecomorph because it has a different climate. But, that remains to be tested. So that's just a picture I want to show you of a colleague of mine holding a giant, crown giant anole. And if you compare that with this, you might imagine that they do have different physiological requirements, indeed. So what's the implications from that study? The implications are mostly, in my view, is when you look at the Lesser Antilles. So this would be Hispaniola again, this is Puerto Rico, and then you have like this whole set of little islands. And I think most of them are inhabited by anoles as well. But there's two things different. First thing is different, they have only 1 or 2 species maximally. And the second thing that's different is that they do not have ecomorphs. So because why are there not many species there? It's because the islands are too small. That's something that John Hills found out in 2000. If you look at this species area curve, you see that like the the largest islands have the most species, and the smaller the island gets, the smaller gets the number of species. So probably there's not enough space to evolve many different species, which could be because there's not many possibilities for allopatric diversification. And also the question why there is no ecomorphs on the Lesser Antilles is, I think we're getting a bit closer to that answer, is because maybe those islands don't provide the appropriate climatic niches that we need on the other ones in order to evolve that whole set that we see. Yeah, and with this I think I'm through. I don't know if I was very fast, I think not. And thank you very much for being here today. And if you have any questions, you can also always write me. Thank you.
Creo que tenemos 5 minutos para preguntas. ¿No tienen preguntas?
I think that correlation could be in the way different places of the islands are affected in different ways of tropical storms, hurricanes or so. So, I mean, several ranges could be like better, less affected of tropical storms, something like that. That's a climate variation.
That might be the reason why Jamaica falls out, because it just has a completely different climate. It was just one example that I was showing, but Jamaica is due to the fact that it's basically sheltered by the other islands, closer to the interior of the Caribbean. Is also, has lots of different temperature parameters. So but that basically, I was sad that Jamaica fell out first, because I would like to have an explanation that involves all of those islands. But the thing is that by falling out and showing us why it falls out, it's actually it's a reinforcement of the pattern that we see in the other ones. So if Jamaica had exactly the same climate and exactly the same ecomorph community complexity distribution, that would be good. But by the way of showing us that because of the different climate, the pattern is different, that tells us that climate is important. That's what I was saying.
Yeah, but I mean, for instance, some ecomorphs are absent in some parts of the island. Could that be related that places are more affected by tropical storms?
Yeah. So basically, we could find that out by correlating the species distribution models with each of the bioclimatic layers, because some of those do reflect the tropical storms. It's basically like the, how do you say, I think seasonality or something. One of those basically tells us how much this area is going to be hit by storms. So I could just pick that one out and correlate the stuff with that one. Yeah.
Okay.
Yeah. So a functional explanation would be definitely worth testing.
¿Otra pregunta?
Yeah.
Well, in the Greater Antilles, there's a tremendous diversity of soil types. You have amazing mosaic of different geologic substrates, and that, of course, has a big influence on plant diversity. And since I work with plants, it's something I know more about. But I was wondering if you see an effect in lizards also, based on different types of substrates, because that affects directly the type of vegetation that you're going to have.
And most likely the way they can stick to that vegetation, right? So that's actually a very hot debate right now on the internet. So because there's a paper that came out that basically proposed to diverge anolis into 8 different genera. And one of the explanations is that you cannot possibly think that you will encounter the same type of structural habitat in all of those cases. So that's an argument to split the genus. But there's been several botanists that have been saying that actually that's not the case, because the type of structure of bark, for example, is not that crazily diverse. You have rough bark, you have smooth bark, but it's all going to be in the same place. There's one tree that has that type and one tree that has that type.
It has some chemical repellent, I don't know.
Anolis don't care.
No.
They're everywhere. No. But actually, I did this, a student of mine did a study in Hispaniola where she tested clinging force to different types of substrate. She did like a glass plate, and she did a pine bark, which is very rough, and she also did a rock, which is very smooth, and she didn't find any. So, I understand. Sorry.
I know you work with some lizards, but I was wondering if you saw some other different organisms that have the same pathogen found on those islands?
On the Greater Antillean, there is some studies about frogs, that you have basically different frog size classes that evolve convergently as well, but there isn't that much done. Like Madagascar was cool because you can just pick from a full set of whatever you want, but the Greater Antillean, no, not so much. And also the diversity is not that crazy. Anolis are very high, but the rest, I think, I don't know, plants maybe. But frogs, for example, yeah, just have a couple species and that's it.
Preguntas? Well, I have one. Talking about the Cybotis group, do you take into account other species that might occur in the same places where you collected the Cybotis group?
Yes, we did. I recorded that, and it didn't come out as having any effect. But that's because the cybotoids, they don't meet each other. They're allopatrically distributed. The only species that is everywhere is the Anolis cybotis. And mostly, like, the microhabitats are so different that you will have cybotis sitting on one patch and the next species sitting on another. So there's no direct, there's very few places where you will find actually the two species on the same place. And I'm very interested in the data from those. I have a couple of collections of genetic material because I've seen animals that look like hybrids. So there could be something going on. And there's also one nuclear haplotype that I found in one of the analyses that I did of another cybotoid species that came out of cybotis, so there must be some introgression as well. But there's not a direct competition on the wide range because they just don't encounter each other very often.
Okay. I'm talking about the definition of the ecomorphs of the Caribbean region. You mentioned at the end of your talk something about the two economics. When you read the Lossus work about the definitions of these ecomorphs, it seems that you always take into account the effect of the body size on body shape, right? But it seems that it's really important to have into account the body size, to delimitate different ecomorphs.
Like, for example, the giant twig anoles, they've been proposed, but those is kind of trash dolls because, the only difference is the size. But apparently it's important because there are a couple of species, for example, chameleons, I don't know if you know those. They're like a huge species from Cuba. I don't know where else they occur. There's another island that also has one of those. It's like a huge anole with short limbs and moving like a chameleon, but it's like this big. And it's not in any of the ecomorph categories right now. But I think at some point somebody's going to propose that giant twig anole should exist.
Yes, and I think that because we have here in the mainlands, we have some kind of similarities with the things in the Phenactosaurus, the Phenactosaurus groups. Actually, there are some species, well, the difference in body size are not so huge, but you can see they are medium size, Anolis, and small ones, even if they have these very short arms.
If you believe my story, and you assume that there's different physiological constraints on each of those, body size has to be important, body mass.
Thanks a lot.